It's possible to classify chorioallantoic placentas based on the way that they attach to the inside of the female's uterus. So the names of the different types are diffuse, cotyledonary, zonary, and discoid. And there's a variation of discoid that's called bidiscoid. This website here has a lot of good information on the different types of placentas and the differences, and nice diagrams that show how these kinds of things look. If you're in lab, you've seen some actual specimens of the different types. But basically, the biggest difference is where the blood vessels are on where they come to the outermost parts of the placenta to make contact with the female's uterus, and what size and shape those places where that increased vasculature is. So probably the easiest to conceptualize is the diffuse placenta. So this is the chorion that we're seeing, and the developing embryo is inside here. And all over the surface of the chorion are little tiny places where there's increased vasculature that can come into contact with the lining of the female's uterus. Then we have the cotyledonary structure, which again, this is the chorion, and the developing embryos are inside here, but on the outside are little knob-like structures that are highly vascularized. And these almost look like buttons, and they fit into little holes that are almost like buttonholes in the lining of the uterus. So there's lots and lots of these different cotyledonary enlargements on the outside of the chorion. Then we have the zonary type of placenta, and here we have the membranes that enclose the developing embryo, and around the middle of each one, almost like a belt around the very middle, is an area where there's a lot of vascular tissue, and that comes into contact with the inside of the uterus. And remember, in species that have lots of offspring, the uterus is sort of elongated, and it may be even doubled. So these embryos kind of line up like peas in a pod. So there'd be one here and another one here and another one here, and all along the way the band, the zonary band around the outside of the placenta, is what's in contact with the inside of the female's uterus. And then finally we have the discoid variety, which has all the fetal membranes and the developing fetus in here, and then there's blood vessels that run to a disc-shaped structure on the very edge of the chorion, and that is what is deeply entangled in the inside of the female's uterus. The bidiscoid situation, there would be two of these, but this is the discoid placenta that's like what humans have. And so if we look what is going on at a microscopic level, where the tissues of the placenta come in contact with the uterus of the female, there's several different types of connections there. And so if we look at the very basic anatomy of that part of the placenta, there are 6 different layers of tissue that can appear between where the fetal blood is circulating and where the maternal blood is circulating. And I want to emphasize here that there's never any mixing of blood between the fetus and the mother. There's always some degree of separation, some layers of tissue in between the two, but how much is in between varies from species to species. So in the kind of most extreme condition, the typical layers of tissue, if we start with maternal blood circulating through the mother's body, then the blood travels through blood vessels that have an endothelial layer of cells that surround, that create those vessels. Then outside of the blood vessels, there's connective tissue in the uterus that the blood vessels are running through. And then finally, at the very edge of the inside of the uterus, there's a layer of epithelial cells. Then the placenta is growing against that. So the very outermost layer of cells in the placenta is the chorion. And then there's connective tissue inside of that. And then finally, there are the blood vessels that have a layer of endothelial cells that make up the blood vessels. And that's where the fetal blood runs. And the same website that we just went to has some really nice diagrams of how that works. So here's the depiction. So you have, starting on the maternal side, you have the maternal blood cells running through the blood vessels. This is a cross section showing the external endothelial cells that make up the walls of the blood vessel. Then there's some connective tissue and a layer of epithelial cells, and then a layer of chorionic epithelial cells, some connective tissue, the blood vessels of the fetus, again in cross section, so you can see this layer of endothelial cells, and then the contents of the blood vessels on the fetal side. So this is our starting point. But when we look at what's going on inside different mammals, some of these layers are not permanent structures. That is, as the placenta starts to work its way deeper and deeper into the uterine lining, the layers of cells kind of get broken apart so that there's more close association between the fetal circulation and the maternal circulation. And that varies from species to species. There's a set of terms that we use to refer to these different types. So there's epitheliochorial, endotheliochorial, and hemochorial. And the trick to understanding what these mean is to understand what the roots of the words are. So chorial always refers to the chorion of the placenta, and in all cases that's present. Then epithelio refers to the epithelium of the female's uterus. Endothelio refers to that layer of cells that makes up the blood vessels on the maternal side. And then hemochorial hemo refers to blood. That's the female's blood. So in the most, I guess, well-developed situation, in all cases, the layers of cells on the fetal side are intact. That is, you can always see the chorion and the connective tissue and the endothelium that makes up the blood vessels on the fetal side. But on the maternal side, the different layers sometimes get eroded away as the placenta works its way in. So in the epitheliochorial, the chorion makes contact with the female's epithelium. That's where epitheliochorial comes from. It's the two layers of cells on the maternal side and the fetal side that are touching each other. The endotheliochorial, as you might guess, is where you have the chorion of the fetus right up against the endothelial cells that make up the blood vessels of the female. So now the epithelium and the connective tissue in the uterus is no longer there. The chorion, the villi of the chorion have eaten their way in so far that they're running right up against blood vessels of the maternal circulation. And then hemochorial is a situation where the chorion of the fetus is bathed in blood from the female's circulation. So here's the endothelium and it's broken open, so the blood vessels are actually just pumping blood out and the blood is running right up against the chorion. So as you might guess, this is the most efficient situation for transfer of nutrients and transfers of gases because everything is happening based on diffusion. The fewer layers of cells there are, the easier it is for substances to diffuse across in either direction. So this is a little less efficient, and this is even a little less efficient than that. Alrighty, so that brings us to um, looking at the evolution of these different patterns of attachment. And so one of the questions we might ask is this all random, or are there particular lineages where you see one type of placenta attachment type or invasiveness type, or is it kind of randomly distributed across mammals? So if we look first at the amount of invasiveness, we've got the hemochorial, endotheliocorial, and epitheliochorial as our three types. And here is our phylogeny. This is based on the Murphy 2001 tree. We have our outgroups here, which are the marsupials, and you can see the marsupials show the epitheliochorial type. That's the type in which all three levels of maternal tissue are still present, and that makes sense if you think about the marsupials not having a really invasive placenta. The placenta is just sort of nestled up against the inner surface of the uterine lining. But if we look at the placental mammals, we see a variety of different types. The most common type is the hemochorial, the situation in which there's been erosion of the uterine lining to the point where there's blood running right up against the chorion of the placenta. And we see some groups that show epitheliochorial structures, and that they're sort of scattered throughout, and others that are sort of scattered throughout that show endotheliocorial. So if you reconstruct this based on what you might predict common ancestors of present-day species to have over time, what you end up with is a pattern where the hemochorial placental type evolved really early in mammalian evolution, somewhere right around the time where marsupials diverged from placental mammals. And we can't really resolve this. We don't know which came first. It could be either one of these two types. And then the other kinds of placenta types are more recent evolutionary innovations. If we look at the shape of the contact zones, the diffuse cotyledonary, zonary, and then discoid or bidiscoid, we see that the discoid placenta type is ancestral, and all the others are more recent evolutionary innovations that happen within particular clades. So the bottom line there is that we as humans tend to show the ancestral pattern of both attachment type and invasiveness. We have hemochorial placentas with discoid attachment.